Our next series of lecture topics are all going to deal with different aspects of cell physiology, and I'm going to be dividing these topics into three main subtopics. The first subtopic that we will explore is called membrane transport, which deals with the question of how things get into and out of cells. Our second topic will be all about the fundamentals of cell energetics, examining how cells harvest energy to get work done. That part might start out feeling a little bit like chemistry and even physics again, but then there's going to be a biological twist. And then third, we'll explore how cells communicate with other cells, or in other words, how cells send and receive signals. And you'll start to see in this series how we're going to build on some of the biochemistry that we've learned so far, and we'll start applying that to living organisms. And we're going to start this series out then by talking about cell membranes and briefly talking about something of a biological puzzle. So here is a fish called an ice fish. So its Latin name or biological name is Chiondraco rastrospinosus, quite a mouthful. And this is an animal that lives in the southern oceans near Antarctica. It's often found very deep in ocean water. But one of the most amazing things about this fish is that this fish can survive and thrive in water temperatures that are really close to freezing, temperatures that would kill you or me. And along with that, on the other end of the temperature scale, this fish starts to overheat and die at temperatures that are anywhere higher than 13 degrees Celsius. So that's 55 degrees Fahrenheit. So temperatures where, we're barely comfortable and cold are so hot that they are a harbinger of death for these fish. So that leads us to a causal question, which is, how is it that some animals like this ice fish can survive in freezing cold temperatures while others die? And the reality is there are a whole bunch of different hypotheses to address this question, but one particular hypothesis where there's some good support is that this has to do with properties of this fish's cell membranes. So now, we've already talked a little bit about phospholipids and also about how fatty acids can vary, and this is a really big clue. If it's possible for fatty acids to vary, so they could be, either saturated or unsaturated, and they can also differ in how long they are, and that variation influences the melting temperature of that substance, then maybe animals that tend to live at either warmer or cooler temperatures might have cell membranes with different fatty acid compositions that allow those membranes to remain liquid, also called fluid, at the temperatures at which those animals live. This is what we actually find. Animals that live at extremely cold temperatures tend to have many more unsaturated fatty acids in their phospholipid bilayers, such as what's here, because those unsaturated fatty acids facilitate fluidity at those temperatures. And in contrast, animals that live at much warmer temperatures tend to have more saturated fatty acids in their phospholipid bilayers, so that they literally will not melt at those higher temperatures. But there's one other surprise ingredient in a phospholipid bilayer that also contributes to the membrane's stability, and that ingredient is cholesterol. So remember that cholesterol is also a type of nonpolar lipid, so that means that cholesterol can hang out in the fatty acid part of the phospholipid bilayer without any problems. And cholesterol's behavior in the phospholipid bilayer is kind of surprising. Instead of actually changing the membrane's melting point, what cholesterol does is it moderates the behavior of the membrane. And what I mean by that is, at higher temperatures, cholesterol just kind of gets in the way and slows down the phospholipids so they don't melt as quickly. And then meanwhile, at lower temperatures, cholesterol still gets in the way, and what it does is it prevents the membrane from solidifying. So based on all of this, this is now probably a good moment to take a minute, pause the video, and write down what you might expect to find in the cell membranes of an ice fish. And so now we're also going to take a look at a couple of other illustrations about how cell membranes vary. So this next drawing here looks really busy. And, I don't want you to worry about memorizing anything that's in this picture. This is just for the purposes of exploring how a cell membrane can vary. So first thing to notice in this picture is that there are different types of phospholipids illustrated. So you can see that there's a cluster of phospholipids here in the middle. And in the middle, there's a whole bunch of cholesterol stuffed in as well, these little yellow bits here. And in contrast, towards the edges, what we find are more unsaturated fatty acids, so unsaturated phospholipids, and not as much cholesterol. So this structure that's pictured here in the middle is actually something that's called a lipid raft in the middle of the cell membrane. And this cartoon also illustrates again for us some of the different kinds of fancy proteins that can be found in cell membranes. So here are some examples of just some cartoons of some of those transmembrane proteins. So trans meaning across. So these membranes go across, or these proteins go across the whole cell membrane. And then there's also some examples of some proteins that stick out either on the outside or on the inside of the cell membrane. And all these types of proteins can have a lot of different functions. So they can be involved in transporting substances, more on that in a little bit. They can act on enzymes, more on that soon. Or they can be involved in signaling. There's going to be more on that soon too. Or they could just be anchor points that help to connect cells together. So now we're going to go through a couple animations that I think are also useful for just seeing how cell membranes work on a cellular scale. So this first one just shows you cholesterol present in one of those lipid rafts in a cross-section of a membrane. So you can see phospholipids in the membrane can all move around quite a lot. They tend to move here in the horizontal plane. They can't move or flip so easily in the vertical plane. So that's another thing to think about. Why would it be hard for a phospholipid to flip in the vertical plane here? But they can wiggle around in this horizontal plane. And then here's one of these lipid rafts, and you can also see some globular proteins. Some of them are transmembrane proteins, so spanning the entire membrane here. Here's an example of two proteins interacting on the surface of a membrane. So again, kind of showing you how these phospholipids on the membrane can move. So one of these proteins is a receptor protein. The other is a signaling protein, so interacting on the surface. And then here's just another cute little example of one of those lipid rafts floating around in the cell membrane. Okay, so that's an introduction to what cell membranes are made out of and how they can vary. And that's going to now set the stage for thinking about this next question of how things get into and out of cells. So maintaining the internal environment of a cell and all of its constituent ingredients requires that a cell membrane perform a complex double function. So a cell membrane has to keep certain substances out while letting others in, and then at the same time it has to keep certain substances in while letting other substances out. And the way that a membrane can accomplish this is going to depend not just on the physical and chemical properties of the membrane itself, so not just the physical and chemical properties of the lipid and protein structure, but it's also going to depend on the physical and chemical properties of the substances that are interacting with that membrane. So think about things like ions, molecules, and aggregations of molecules. But before we really dive in there, we also need to take a moment to talk about two principles. And the first principle we're going to talk about here is diffusion. And hopefully from the lecture video on endosymbiosis, you've got a head start on thinking about this process. But just in case, here's a definition of diffusion. So diffusion is the movement of atoms or molecules from an area of higher concentration to an area of lower concentration. And I'll show you an illustration of that here. So here's a fun animation of diffusion at work. So all these molecules or atoms have started out at a higher concentration initially. This will replay again. And through time, through random movement, those atoms or molecules diffuse and redistribute until they reach a lower concentration. So if we're, here's a picture, a little cartoon I drew of some water where the initial concentration of sodium, so symbolized here, is higher on the left, and the initial concentration of water, these little red molecules, is higher on the right, and so therefore the concentration of sodium is lower. So if we're thinking in terms of diffusion of both of the substances that are present, sodium is going to diffuse towards the right in this image, and water is going to diffuse towards the left. And we're going to revisit this idea again momentarily. So diffusion's our first principle. Our second starting principle is the principle of selective permeability. And this is just the idea that not everything can pass easily through a membrane. And again, there's some basic chemical principles that are going to determine what can and cannot pass through a cell's phospholipid bilayer. And to just give you a hint about this, it has to do with whether the substances are charged, polar, or nonpolar. We're going to get back to that in a minute, but first we're going to take a look at a couple of examples of how the presence of a selectively permeable membrane affects the movements of solutes and water across that membrane. So for this initial practice, we're going to be working with a synthetic type of membrane. It's actually a type of membrane that's used in kidney dialysis machines. So if you've ever known anyone that's had to be on one of these machines because of trouble with their kidneys, this is what's filtering their blood instead of their kidneys. So this membrane type is often called dialysis membrane. And this membrane has some properties that make it very different from a phospholipid bilayer, so be very careful to distinguish between the two depending on the context. So here we've got synthetic membrane. So this dialysis membrane is semi-permeable. So it's basically a really, really fine mesh, and so what that means is that small molecules such as sodium and water can travel through it freely, while bigger molecules like glucose can't fit through. And given these properties, I'm going to show you a couple of scenarios and then talk about what will happen in each scenario. And so here's our first scenario where the starting concentration of sodium is high on the left and lower on the right. And again, since this membrane is permeable to both sodium and water, the result we will see will be the same as for that first example without the membrane, right? So sodium is going to diffuse to the right, and water will diffuse to the left. So that's pretty simple. And by the way, there are names for these starting conditions when there's a membrane involved. So these conditions are referred to as being hypertonic. So hyper meaning more, and tonic referring, this is kind of like salty. So, more dissolved substances here on the left, and hypotonic. So hypo meaning less, so fewer dissolved substances on the left. Okay, so that's pretty simple. So now we come up to scenario number 2. So here we're dealing with glucose rather than sodium, and the glucose concentration is higher on the left. So now glucose can't diffuse onto the right side because this synthetic membrane is not permeable to glucose. However, water can still move, so water is going to diffuse to the left. And this is what we're going to wind up with, something like this. Okay, and now what about if there are equal concentrations of glucose on both sides? In this case, there's going to be no net movement. Okay, and so the water is still going to move around, glucose is still going to be trapped, but there's going to be equal amounts of water moving to the left and moving to the right. And so in a situation like this, where the concentration of dissolved solutes is equal on both sides, that's referred to as isotonic. So iso meaning same. Okay, so that's a hypothetical situation. And now we need to think about how that's going to translate into thinking about the movement of substances into and out of cells. And broadly speaking, we can categorize the movement of substances into and out of cells based on whether the substances are able to diffuse down their concentration gradient or not. And so if substances are indeed able to diffuse down their concentration gradient, so they're moving from one side of the membrane to another, diffusing down, that process is referred to as passive transport. And there are four major forms of passive transport that we're going to talk about here. The simplest form, simple diffusion, simple, is what happens for small non-polar molecules. So these are molecules that can freely cross through the phospholipid bilayer because they're non-polar. So that's all they need to do, just diffuse down their concentration gradient. The remaining three forms of passive transport here are forms that all involve some type of protein or another. And so they are categorized based on the type of protein that's involved and what that protein moves. So these are all generally referred to as carrier-mediated or protein-mediated forms of transport. So stuff can't get across the membrane directly. It needs some kind of mediator or facilitator to help out. So let's look at these types in detail. So the first of these are the ion channels. So here's an example of an ion channel protein. And this is the ion channel protein for sodium. And channel proteins typically have a water-filled pore, pictured here, in the middle. But the pore is usually specialized to be selective, as I note here, and only let specific types of ions through, as determined, as I note here, by the pore's size and its charge. So I want you to take a moment to think about another question, which is if the amino acids lining a pore are positively charged, what types of ions do you think are more likely to pass through? Cations that are also positively charged, or anions that are negatively charged? So if you guessed anions, you are correct. So that's ion channel transport. So again, a water-filled pore. The next type of carrier-mediated transport, passive transport, is called facilitated diffusion. And at first, the facilitated diffusion might look a lot like an ion channel, but there's an important difference, which I'm trying to illustrate here, and that is that there's never an open channel present in one of these proteins. So instead, these proteins actually change shape as the cell membrane bends and flexes, and as it does so, in this case, if the right type of molecule comes along, it can bind to the carrier protein, and then as the protein flexes and changes shape, then that molecule can get released on the other side of the membrane. And so again, these never create a continuous passageway. And this type of carrier protein will often transport things such as glucose or simple amino acids. Okay, so then our final type of passive transport protein is one of my favorites. And I think it's one of my favorites mostly just because these were only actually discovered about 30 years ago, in spite of the fact that these proteins are one of the most important transport proteins ever. And so what I'm referring to here are proteins that are called aquaporin proteins, which allow for the simple diffusion of water, or I should say carrier-mediated diffusion of water. And now, because most cells have a ton of aquaporin proteins in their membranes, cells often behave as though water can just diffuse freely across the membrane itself, but water cannot. So it has to travel through one of these aquaporin proteins. And the picture here is an illustration of what this looks like on a molecular level. So these yellow things here are the polar phospholipid heads, and then the green stringy stuff are the fatty acid tails. And the blue ribbons running through this membrane are the aquaporin protein, and then these little red blobs are water molecules. So you can see water molecules funneling through one of these aquaporins. Okay, so now we're going to come to a more theoretical sort of question, which is the question, what are some ways to tell whether a molecule is crossing a membrane using simple diffusion versus facilitated diffusion? So one method for telling is to look at the relationship between the concentration gradient, so the relative amounts of how much stuff is on one side versus the other, versus the speed at which stuff moves across that membrane. And the fancy term for that is flux. Okay? So, and here's the key giveaway. So if something is moving by simple diffusion, then the rate at which it will cross the membrane is going to always be directly proportional to the concentration gradient. So here as the concentration gradient increases, the flux also increases linearly, right? Whatever that concentration is, there's always an increase in flux as that gradient increases. But then in contrast, for facilitated diffusion, or carrier-mediated transport, there's actually going to be a maximum point where even if this concentration gradient keeps increasing, the rate of movement into or out of the cell can't increase anymore. And I think a good way of thinking about this is to think about a crowd of people trying to leave a building. So if you have a small crowd and you have a whole bunch of doors, you've got no problem. As the crowd size inside the building starts to increase, at a certain point there's only going to be so many doors in that building, and that's going to limit how quickly people can leave the building. And eventually, the departure rate is going to hit a maximum, and it can't go any higher than that. There's only so many exits from the building. And so that point of maximal flux, shown here, is also referred to as the saturation point for those carrier proteins. They're saturated and they're full, they're clogged, they physically cannot transport anything faster than that. Okay, so that takes us through passive transport. So moving things from higher concentration to lower concentration. So allowing things to diffuse down the concentration gradient. But now, what if you want to bring something in against its concentration gradient? In that case, some energy is going to be required because this involves fighting against the laws of physics. So it's kind of like rolling a rock up a hill. So the forms of transport that involve moving substances against their concentration gradient are referred to as active transport. And so these methods all require some form of energy, typically in the form of energy that's stored in the bonds of a molecule known as adenosine triphosphate, or ATP here. And we're going to talk more about ATP pretty soon. So to give you an example of one of these active transport methods, here's an example of what's also called a protein pump that moves both sodium and potassium against their concentration gradient. So this is called a sodium-potassium pump. So to summarize what's going on in this cartoon, the sodium concentration is indicated here, and so sodium concentration is higher here outside of the cell. So this is called the extracellular space outside of the cell. And then meanwhile, the potassium concentration is higher here on the inside of the cell, so in the intracellular space. And now, so the way that this transport protein works is so sodium will bind to this transporter, and then at some point a molecule of ATP will bump into this transporter, and it's going to release energy that's going to trigger a change in the shape of the transporter that will release 3 sodium ions outside of the cell. And then while this protein is in that changed shape, 2 potassium ions can bind. And then lastly, a phosphate group will come free from the transporter, causing it to change its shape back again and release 2 potassium ions inside of the cell. So there's an exchange here. So it takes energy and it pumps 3 sodium out, and at the same time it will pump back 2 potassium ions back into the cell. And now, if we wanted to get really technical, there's a name for this particular type of active transporter. This is called an antiporter because it moves 2 things in opposite directions from each other. And that's in contrast to some transport proteins that move 2 things or more than 1 thing in the same direction. Those are referred to as symporters. But antiporter and symporter are more advanced topics for the moment. You'll dive into more of that further on. I do want to note one other really cool case of active transport, which is accomplished by the protein that's illustrated here, which is the active transport of hydrogen atoms, aka protons, against their concentration gradient. What I want you to think about is this question here. If a cell only moves positive hydrogen atoms in one direction across the membrane, what do you think will happen to the electrical charge on either side of this membrane? So it's going to change, right? Right, it will. And so what will happen is there will start to be not just a chemical or concentration gradient, but also an electrical gradient across the cell membrane. And as an added level of complexity, the direction that any given ion will travel is determined by both the concentration gradient and the electrical gradient. And this is also a good place to point out that most of the positively charged ions that are found in animals like us are not in chemical equilibrium among the body compartments. And as I note here, this is something that will get explored a lot further in later courses, but I want to introduce the idea here. So instead, there are concentration and electrical gradients across most membranes, with higher concentrations of sodium and potassium ions outside of cells, and then higher concentrations, actually potassium's higher inside of a cell. So higher concentration of sodium outside of a cell, higher concentrations of calcium and potassium inside of cells. So what this means is that we're all basically like a biological battery. So we're full of a ton of chemical and electrical potential in our cells. Okay, so that takes us through direct or primary active transport with ATPases. And we're going to leave out some more complex forms of active transport. Those are, again, topics for later courses. So the main thing that's left for us right now are the transport categories that are outlined over here, which are referred to as bulk transport methods, because they each involve moving a larger amount of something into or out of a cell. We've actually already watched an animation for one of these forms of bulk transport. So for exocytosis, which is the process by which a cell releases substances into the external environment. And so this is the type of process that's used by cells called secretory cells, so cells that secrete or release things to export products to the environment outside of a cell. So this includes things like the insulin that gets released by the pancreas into the bloodstream, or the release of some neurotransmitters by neurons. So that's pictured in the cartoon here. So a nerve impulse comes. We've got a bunch of vesicles here that contain a neurotransmitter molecule. When the signal arrives, that triggers these vesicles to fuse with the membrane and dump their contents out into this space in between cells. Okay, and the same can be true of other molecules released into the extracellular space. Okay. So that's exocytosis, so release. I like to think of it as exo sounds like exit. So release of substances through bulk means. So the reverse of that is then called endocytosis. So endo meaning into. And so that's going to be the bulk movement of substances into a cell. We also actually have already seen an example of this with the endosymbiont hypothesis. So endocytosis, endosymbiont, where bringing in a solid object such as another cell is a process referred to as phagocytosis. That's just a fancy word for cell eating. So here's another scanning electron microscope image of freshwater denidium cell eating a paramecium. There's also a separate term for endocytosis when it involves liquids. It's called pinocytosis. But at that point, that's just an excess vocabulary word. So I wouldn't get too bogged down by the vocabulary so much as understanding that it's possible for cells to take in bulk quantities of things from their environment through endocytosis. So this picture here shows some capillary cells, so along the edge of a blood vessel, that are actually taking up liquid from the blood into the capillary cell. So a way of getting that liquid into the cell. Okay, so that is our overview of how stuff can move into and out of cells, membrane transport. And so, just to recap again, things can move passively down their concentration gradient, either through simple diffusion or by protein or carrier-mediated transport, or substances can be actively transported against their concentration gradient, either through some form of protein pump that uses ATP for energy or in bulk forms. And so now up next, we will continue moving forward by talking more specifically about cell energetics. So here we're going to look at what the big deal is with ATP and how cells harness energy to get work done. So from here, onward.